Welcome to the Barrier Breakdown, Disrupting Mental Health podcast, where we talk about the clinical and practical issues that face those working in the mental health industry.
Hello, everyone, and thank you for joining us for this week's episode of the Barrier Breakdown. My name is Erin Molyneux-Bailey. I'm the Chief Operating Officer at Cognitive Behavior Institute, and my co-host, CEO and owner of Cognitive Behavior Institute, Dr. Kevin Caridad. On today's episode, we are discussing an experimental vaccine that could help fight the opioid epidemic with Dr. Sandra Comer. Dr. Comer is a professor of neurobiology in the Department of Psychiatry at Columbia University. She is the director of the Opioid Laboratory in the Division on Substance Use Disorders, and her current research focus is on the clinical testing of medications for treating opioid use disorder, methods to maximize the use of naloxone by opioid users, and evaluations of the comparative abuse liability of prescribed pain medications. So thank you so much for taking the time to be here with us, Dr. Comer. And can you start the conversation for our listeners? We'd like to start off by learning a little bit on, how did you become interested in studying opioids?
Oh, sure. So I guess my interest in studying opioids started in graduate school, where when I started, I was in the biopsychology program at the University of Michigan, and my mentor was Dr. Jim Woods, who's a really well-known behavioral pharmacologist. And at the time, there were three subtypes of opioid receptors. There's mu, delta, and kappa. And mu is the receptor that, a lot of the abuse drugs act that we know about, heroin and oxycodone and now fentanyl. But we were interested in trying to find an analgesic, so a pain medication that would provide effective pain relief, but would not have the abuse potential of, opioids like morphine. So I was, tasked with trying to characterize the behavioral pharmacology of the delta subtype of opioid receptor. So, fast forward now, my interest in opioid pharmacology just continued throughout this entire period. And unfortunately, we have gotten to the place where we are now, where there was a lot of overprescribing of pain medications, and now there's this huge population of people who are suffering from opioid use disorder.
Yeah, no, that's absolutely correct. And can you tell us a little bit about the concept of a vaccine and how that comes into play?
Sure. So the concept with the vaccine is not entirely new. I did my postdoctoral fellowship at the University of Minnesota, and one of the investigators there, Paul Pintel, had been developing a vaccine that was targeting nicotine, to help with cigarette smokers. So, and then not too long after that, another group developed a vaccine for treating cocaine use disorder. So the idea is that the vaccine would target a particular chemical structure. So in the case of nicotine, or for cigarettes, it would be nicotine, and for cocaine, it would be cocaine. And so the vaccine is given to a person who would generate antibodies that would target that chemical structure. And if someone, smoked a cigarette or used cocaine, then the antibody would bind to that molecule and prevent it from getting into the brain. So the opioid vaccine is conceptually the same. So the person would get vaccinated over a period of time. They would generate antibodies to the opioid. And if someone chooses to use the opioid again, then the antibody would bind to the opioid, and it becomes a large complex, and it won't cross into the brain.
Just stops the dopamine release to it. No kind of positive benefit from it, it sounds like.
Yeah. And my interest in starting to study this just kind of happened by accident. I saw my colleague Paul Pentel at a conference and just sort of ran into him, and I said, "So, hey Paul, what are you up to these days? Are you still working on your nicotine vaccine?" And he said, "Yeah, we are, but we're also developing this opioid vaccine." My first reaction was, why do we need an opioid vaccine? I mean, we have all these great medications that are, really useful for treating opioid use disorder, including buprenorphine and methadone and naltrexone. And I was just like, I don't understand what the value of an opioid vaccine would be, because, it just seems like a strange concept. So the advantage actually of an opioid vaccine is there are multiple potential advantages. One is that we're hoping that it lasts for a long time. So, once somebody becomes vaccinated, the antibody levels will remain sustained for several months, if not longer. And that is not the case with any of the existing treatment medications. So we have sustained release formulations of buprenorphine and naltrexone. The buprenorphine is a partial opioid agonist. Naltrexone is an antagonist or an opioid blocker, and they last for up to a month. There are some formulations of buprenorphine that can last for longer than that, but not quite as long as we're thinking that a vaccine might last. So that's one of the advantages. The other is that with trying to get a patient onto, especially naltrexone, you have to completely detoxify somebody who is opioid dependent. So, a regular heroin user or oxycodone user has to be weaned off of those medications, and then the naltrexone would be started. And that's a really difficult thing for a lot of patients to do. Buprenorphine as a partial agonist is also a little bit difficult to transition onto the medication, because if you don't do it right, it can make a patient really sick. So the vaccine theoretically doesn't have that problem. The antibody levels will slowly increase, and at least in the studies that we've done so far in rats, that doesn't seem to be an issue. So those are kind of a couple of big advantages.
Now, can you speak more about the research and some of the, so that when you talk about a vaccine and methodology, I think a lot about COVID. The method of the current one is RNA and kind of lasts, they're finding out only so months. Is it similar? Is this a virus-driven kind of vaccine that's then getting the immunoresponse? So what is triggering the immunoresponse? What's kind of the molecule, chemical, or I'm not know if the research terminology as the piece that's really doing the magic?
Yeah, so the vaccine is composed of different parts to it. So there's a hapten, which is chemically very similar to oxycodone, and that's the part that the body recognizes as needing to target. There's a carrier protein that it's connected to. The one that we're using is called KLH. It's keyhole limpet hemocyanide. And then there's another part of it that's called an adjuvant that kind of boosts the immune system. And the one we're using is called alum, aluminum. And alum is a very kind of common adjuvant that's used in vaccines. So it's not similar to the, COVID vaccines that you were just mentioning in that way, but it's similar to some of the older vaccines that have been used clinically. So the idea is that the body would generate an antibody that would specifically target the opioid that is part of the hapten. The thing that's a little bit different with the opioid vaccine idea in contrast to something like the cocaine vaccine. So, the vaccine is targeting a particular chemical structure. With cocaine, that's fine because people use cocaine, it's one chemical structure, and the antibodies would target that structure. With opioids, the chemical structures can be very different from one opioid to another. And even though they all kind of act at the new opioid receptor to produce their effects, the chemical looks different. So oxycodone looks different from heroin, which looks very different from fentanyl. So with this approach, we kind of knew from the beginning that we would need to take a multivalent approach. So that's what we're doing. We're developing an oxycodone vaccine, we're developing a heroin vaccine, and a fentanyl vaccine. And the idea being that ultimately we would give, all of the vaccines together to a patient to help them with their disorder. So that's part of it. That's the downside of this opioid vaccine approach, is we have to take this multivalent strategy. It's also an advantage in the sense that because it's so selective for the chemical structure, then it doesn't cross-react with other opioids. So for example, it doesn't cross-react with methadone or with buprenorphine or naltrexone. So theoretically, somebody can be maintained on one of the existing treatment medications that we know works, and then they can be vaccinated on top of that and provide additional, protection from, experiencing the euphoric effects, which is what we're trying to avoid, because that we know that will lead to increase the likelihood that they'll relapse. And then also the thing that's really nice is that for all three of the currently approved treatment medications, after about 6 months of treatment, about 50% of the patients will relapse, just over the course of treatment. So we're thinking that if we vaccinate a patient who's on buprenorphine, for example, they relapse after 6 months, hopefully the antibody levels will still be circulating and doing their thing. At the very least, it could prevent, somebody from overdosing during that time period. And it can provide us maybe with a window of time that we can re-engage someone in treatment.
So this medication specifically, or I should say the vaccine, it kind of does what it needs to do, so not to have the euphoric feeling, but it also blocks, in a sense, or inhibits the sedative or parasympathetic response.
Yes, because those are centrally mediated, and because it doesn't allow the opioid to get into the brain, all of the effects that are produced by the opioid should be equally affected. So, the worry that we have, clinically, is one of the most worrisome is the, respiratory depression from an overdose.
Sure.
Yeah.
Okay, great. I was wondering. Go ahead, Erin.
Go ahead, Kevin.
With regard to, I could, the excitement of hearing something like this is, you look forward to seeing the studies as an adjuvant. Does it help, which I'm sure you're excited about, does it improve outcomes by the addition to? Is it a standalone? There's a lot of exciting things to see what comes with this, and I wonder, the cost ultimately, and even beyond the U.S., right? Those, is this a costly vaccine? Because often we've had someone on from Canada about a specific medication, and his research is looking at drugs outside, typically in the U.S., because they're not necessarily cost effective in other countries. What does this look like, or we're not even to that point?
We're really not even at that point yet. We're still manipulating the vaccine itself. I mean, we're testing it in people right now. We started with the oxycodone vaccine. Because at the time when we were making this decision, oxycodone, just numbers wise, was the most prevalent opioid that was being used. Things have shifted, over time, and now fentanyl is very commonly used, and that one is seems to be a lot more toxic than the other opioids, because you look at the fentanyl related overdose deaths, and that's the most prominent substance that's found in people who have died from an overdose. So we started with oxycodone, and we're trying to optimize the effectiveness of it, and we're collaborating with a group led by Jay Evans at the University of Montana, who has been developing different adjuvants. And like I mentioned before, the adjuvant that we're using now is alum. But he has a whole bunch of other adjuvants that may be more effective than alum in generating a good immune response. So we're still, we're in clinical testing, which is great, but we're still haven't completely finalized the components of the vaccine. And just want to mention too, because I realized that I didn't say this earlier, and I should have, but the investigator who is really leading this project is Marco Pravatoni. He is a professor in the University of Washington, and he's the one who trained with Paul Pintel, the investigator who developed the nicotine vaccine. But Marco has taken over the whole program, and because Paul retired, and has done all of the development of the vaccine, all the preclinical testing of it in rats, and he's just really, really great. And so wanted to give credit where credit is due, for sure, in that regard. And the thing that I also wanted to mention as being really important in this approach is that, so the nicotine and the cocaine vaccines that I mentioned earlier, they both made it to phase 3 testing. So it was kind of like fairly late stage clinical testing, but both of them ultimately failed because there was a lot of variability across in between subjects or patients who, in terms of their immune response to the antibodies or to the vaccines. So, some of the patients who received the nicotine and the cocaine vaccines generated a really strong immune response, so their antibody levels are very high. Other patients had very modest or mild antibody response. So when the clinical data were evaluated as a whole, there was no significant difference from the control placebo condition, right? So both failed in phase 3 testing. But we know that of the top third of the patients in both of the vaccine studies that generated the really high antibody levels, they did show a good clinical response. So conceptually, it was successful, right?
Sure.
So one of the things that we're doing, and this is something that Marco has demonstrated in his rats, is we're looking for a biomarker that can kind of tell us which patients might be good responders to the vaccine. So what Marco has been focusing on in his rat studies is B cell activity, right? Because that's important to the immune response. And what he has been able to demonstrate in the rats is that the animals that had high B cell activity prior to vaccination were also the animals that showed a really strong immune response. So there was a really nice, significant correlation there. Okay. So we're looking at B cell activity in our research participants that are getting the vaccine. So we've gotten B cell, we're measuring B cell activity before vaccination and then at various points afterwards. And we're hoping to see the same kind of correlation that Marco saw in his rats. So if it turns out that we can find a really reliable biomarker, then that means that once we get to phase 3 testing, we can, pre-screen patients who might be good responders based on their B cell activity. And so basically we can enrich our population with the patients who we feel might be really good responders to the vaccine, and hopefully show that there's a significant difference between the control and the active vaccine.
That'd be like designer vaccine. You're getting really specific.
Mm-hmm. Mm-hmm.
Oh,
Yeah.
great.
So it's very much like individualized or precision medicine, that we're kind of going for.
Real biohacker.
Exactly.
Awesome.
Wow. No, I'm sure a lot of therapists and mental health clinicians who are listening today are, really interested in this being an additional piece, for the clients that they work with, which could really, help change a lot of lives. So that's really incredible, the work that you're doing.
Yeah, thank you.
Oh, thank you. Yeah, we're very excited about it because we think it's very promising, and it has it has a lot of interesting clinical applications, not only for people who are currently experiencing opioid use disorder, but theoretically it can be used for as a preventive approach. So if you think of somebody who might be at very high risk of developing the disorder, that might be good for them. There's potential for use among first responders. We know that drugs like fentanyl and carfentanil have been used in in combat kind of like situations. Were you guys familiar with the Chechen rebels that held a theater, people in a theater hostages in Moscow? Yeah. And so the authorities decided to use this gas to immobilize the hostage takers, the rebels. Yeah. And unfortunately, of course, they also anesthetized the hostages. And the idea was that they would have naloxone or Narcan to reverse the overdose. But unfortunately, quite a lot of people died in that situation. But it demonstrated actually that you can use carfentanil in this way as to cause mass casualties. So you could potentially vaccinate the soldiers or military personnel to help protect them against this possibility.
It's a gas mask without the gas mask.
Right.
Straight out of the show 24, I feel like.
Yeah. Quite the defense medication.
Jack would have really liked that. Yeah.
Yeah.
Wonderful. Well, thanks so much for your time. This was really interesting, and we look forward to continuing and to follow your research and see what the outcomes
Come back
are.
once you're concluded. Yeah, I'd like to hear the
results. We definitely would love to do that, and just want to also acknowledge the National Institute on Drug Abuse for funding this research. It absolutely would not be possible without their support. So
keep funding.
Thank you.
And thanks so much to our listeners for this week's episode of The Barrier Breakdown. And we thank Dr. Sandy Comer for joining us. And we hope you all stay safe and healthy. Take care.
Thanks for listening to The Barrier Breakdown, Disrupting Mental Health podcast. Check out our website at cbrcenterforeducation.com for more information and to learn about upcoming continuing education events.